Extraneal icodextrin PD solution can be used in a wide range of PD patients. The safety and effectiveness in pediatric patients have not been established. It is, in fact, the only non-glucose peritoneal dialysis solution available in the United States. Let's first review how PD works with a glucose-based solution. The capillaries within the peritoneal membrane contain numerous water channels and small and large pores that allow for water movement across the capillary wall, as well as movement of small and larger solutes. Removal of toxins is driven by the concentration gradients between blood and infused solution. Through the process of diffusion, small molecules move from the area of higher concentration in the blood to the area of lower concentration in the solution. Additionally, osmotic agents in PD solutions, such as glucose, which are in hyperosmolar concentrations, generate ultrafiltration that moves water across the membrane. This results in the convective transport of solutes from the blood into the solution, also known as solute drag. The peritoneum thus acts as a filtering device, similarly to kidneys or a dialyzer. These filtration processes continue while the solution dwells in the peritoneal cavity for a certain number of hours. The amount of toxin and fluid removal depends on the type and concentration of the solution, the length of dwell time, and the permeability of a patient's peritoneal membrane. When using a glucose solution during the long dwell, the concentration of glucose may reach equilibrium with the blood. This can result in the loss of osmotic gradient and can lead to reduced or net negative ultrafiltration. The choice of osmotic agent and osmotic concentration should be selected to meet the needs of the individual patient. A patient's volume status should be carefully monitored to avoid hyper or hypovolemia and potentially severe consequences, including congestive heart failure, volume depletion, and hypovolemic shock. An accurate fluid balance record must be kept and the patient's body weight monitored. Now, let's consider how Extraneal PD solution works. Extraneal PD solution induces ultrafiltration by colloid osmosis throughout the long dwell, 8 to 16 hours. Water is transported across the small intercellular pores of the peritoneal capillary endothelium. Icodextrin has a similar osmolality as blood, but its large molecular size induces the movement of fluid by the colloidal osmotic pressure. Icodextrin is a large molecule and is slowly removed from the peritoneal cavity via the lymphatic pathways. As a result, icodextrin molecules remain in the peritoneal cavity for long periods of time, allowing for sustained ultrafiltration during the long dwell. In a single-dose pharmacokinetic study using Extraneal, a median of 40%, 60 grams, of the instilled icodextrin was absorbed from the peritoneal solution during a 12-hour dwell. Like other peritoneal dialysis solutions, Extraneal PD solution also contains electrolytes to help normalize electrolyte balance and lactate to help normalize acid-base status. Overinfusion of peritoneal dialysis solution volume into the peritoneal cavity may be characterized by abdominal distension, feeling of fullness, and/or shortness of breath. Treatment of overinfusion is to drain the peritoneal dialysis solution from the peritoneal cavity. Extraneal is indicated for a single daily exchange for the long 8 to 16-hour dwell. During continuous ambulatory peritoneal dialysis, where exchanges are performed manually, the long dwell is usually during the night. During automated peritoneal dialysis, where an automated cycler assists the patient by automating a number of exchanges, the long dwell usually occurs during the day. Extraneal icodextrin PD solution is also indicated to improve, compared to 4.25% dextrose, long dwell ultrafiltration and clearance of creatinine and urea nitrogen in patients with high average or greater transport characteristics as defined using the peritoneal equilibration test. Extraneal PD solution is an important part of Baxter's comprehensive portfolio of home dialysis products and services. Extraneal icodextrin PD solution indications and important risk information, IRI. Indications: Extraneal icodextrin is indicated for a single daily exchange for the long 8 to 16-hour dwell during continuous ambulatory peritoneal dialysis, CAPD, or automated peritoneal dialysis, APD, for the management of end-stage renal disease. Extraneal is also indicated to improve, compared to 4.25% dextrose, long-dwell ultrafiltration and clearance of creatinine and urea nitrogen in patients with high average or greater transport characteristics, as defined using the peritoneal equilibration test, PET. Important risk information. Warning: unrecognized hypoglycemia resulting from drug-device interaction. Only use glucose-specific monitors and test strips to measure blood glucose levels in patients using Extraneal icodextrin peritoneal dialysis solution. Blood glucose monitoring devices using glucose dehydrogenase pyrroloquinoline quinone, GDH-PQQ, or glucose dioxidoreductase, GDO-based methods must not be used. In addition, some blood glucose monitoring systems using glucose dehydrogenase flavonoadenine dinucleotide, GDH-FAD-based methods must not be used. Use of GDH-PQQ, GDO, and GDH-FAD-based glucose monitors and test strips has resulted in falsely elevated glucose readings due to the presence of maltose. Falsely elevated glucose readings have led patients or healthcare providers to withhold treatment of hypoglycemia or to administer insulin inappropriately. Both of these situations have resulted in unrecognized hypoglycemia, which has led to loss of consciousness, coma, permanent neurological damage, and death. Plasma levels of Extraneal icodextrin and its metabolites return to baseline within approximately 14 days following cessation of Extraneal icodextrin administration. Therefore, falsely elevated glucose levels may be measured up to 2 weeks following cessation of Extraneal icodextrin therapy when GDH-PQQ, GDO, and GDH-FAD-based blood glucose monitors and test strips are used. To avoid improper insulin administration, educate all patients to alert healthcare providers of this interaction, particularly in hospital settings. The manufacturers of the monitor and test strips should be contacted to determine if icodextrin or maltose causes interference or falsely elevated glucose readings. For a list of toll-free numbers for glucose monitor and test strip manufacturers, please contact the Baxter Renal Clinical Helpline at 1-888-RENAL-HELP or visit www.glucosesafety.com. Because of the risk of unrecognized hypoglycemia that could result from a drug-device interaction, Extraneal is available only through a restricted program. Extraneal icodextrin is contraindicated in patients with a known allergy to cornstarch or icodextrin, in patients with maltose or isomaltose intolerance, in patients with glycogen storage disease, and in patients with severe lactic acidosis. Extraneal is intended for intraperitoneal administration only, not for intravenous injection. Aseptic technique should be used throughout the peritoneal dialysis procedure. Encapsulating peritoneal sclerosis, EPS, sometimes fatal, is a complication of peritoneal dialysis therapy and has been reported in patients using Extraneal. Serious hypersensitivity reactions to Extraneal have been reported, such as toxic epidermal necrolysis, angioedema, serum sickness, erythema multiforme, and vasculitis. Anaphylactic or anaphylactoid reactions may occur. If a serious reaction is suspected, discontinue Extraneal immediately and institute appropriate therapeutic countermeasures. Overinfusion of peritoneal dialysis solution volume into the peritoneal cavity may be characterized by abdominal distension, feeling of fullness and/or shortness of breath. Drain the peritoneal dialysis solution from the peritoneal cavity to treat overinfusion. Patients with insulin-dependent diabetes may require modification of insulin dosage following initiation of treatment with Extraneal. Monitor blood glucose and adjust insulin if needed. Peritoneal dialysis may affect a patient's protein, water-soluble vitamin, potassium, sodium, chloride, bicarbonate, and magnesium levels and volume status. Monitor electrolytes and blood chemistry periodically. Monitor fluid status to avoid hyper or hypovolemia and potentially severe consequences, including congestive heart failure, volume depletion, and hypovolemic shock. Abnormalities in any of these parameters should be treated promptly under the care of a physician. In clinical trials, the most frequently reported adverse events occurring in greater than or equal to 10% of patients and more common in Extraneal PD solution patients than in control patients were peritonitis, upper respiratory infection, hypertension, and rash. The most common treatment-related adverse reaction for Extraneal PD solution patients was skin rash. Please see accompanying full prescribing information. With over 30 years of home therapy experience, Baxter is committed to supporting patients and clinicians to help ensure that home works.